Let's try now.
Okay, can you hear? Everybody good? Sound coming through? Should I continue or?
Okay.
Okay, all good, all right. So again, we're interested in how cells communicate in wounds, particularly different inflammatory cells. And we're looking at different methods to perturb the system and improve healing, including biological and mechanical approaches, and kind of trying to manipulate cells and introduce them into the wound to improve healing. So get to this slide to acknowledge the people that actually do the work in the laboratory. I have three very talented people at different levels here: a postdoc, research assistant professor, and a lab manager that listed on top: Jingbo, Jacqueline, and Rita. I work with three different informatics groups, computational biology groups, most notably Ying Dai, who's in the engineering department, and a couple of her students. They're very important in kind of helping us analyze our bigger data sets. A new faculty, Matt Witeka, also kind of invested in kind of using informatics approaches to understand the healing process. So these are the people that do most of the work. I just get to help direct them and try to interpret all the data that they generate.
Always a new challenge.
Where is it? There we go. Okay. So the problem we're dealing with are these chronic wounds. They afflict, at least in the US, over 6 million patients. This is a problem around the world. Anywhere where diabetes is a problem, treatment is very expensive. These wounds tend to recur. So even though you can heal the wound, tends to come back. These wounds, very ugly looking things here. Going to the wound clinic personally was an experience. And if these aren't taken care of, often lead to amputation. And once you end up with an amputation, you're on this path to more amputations, and mortality rates are rivaling cancers. So people die from wounds, lots of people, and this is not something that's well known. The causes still are less known, and we are looking for effective treatments. Usually it's just cleaning, putting on dressings, but we're trying to come up with different methods to improve healing. In our case, targeting that inflammatory response. There we go. So wound healing, so I know most of you don't think about wound healing very much, except for most of you, if you get a wound, you'll heal very quickly and effectively. But people that don't heal well, it's this big problem. We think of this occurring in three phases. First one is called the inflammatory phase, where inflammatory cells like neutrophils and macrophages accumulate in the wound. They clear out the damaged tissue and help prepare the wound for healing. The next phase is called the proliferative phase. So in that phase, you get proliferation of fibroblasts, other cells that help to form the new tissue. And then finally, there's a remodeling phase, where the wound is remodeled to resemble the tissue that was there before wounding. But usually, especially with skin, you end up with a scar. It doesn't ever come back to gain its full function again. And in the case of wounds in diabetic patients that don't heal well, they're often described as being stuck in this inflammatory phase. So the inflammatory phase does not resolve, and you don't progress to these later phases in the healing. Still having a problem finding it. Okay. So I'm going to talk, this is an overview of the talk. We'll go through heterogeneity of cells called monocytes and macrophages in normal and impaired healing. Next, we'll cover communication between monocytes and macrophages and other wound cells. We'll get to then impact of mechanical stimulation on these cells and their communication. And maybe if we have time, we'll get to the part about communication between different organs in the body, particularly in these data between the liver and the skin. That's important for healing. That has not been appreciated. So first of all, this is a topic, kind of a main focus of my lab, trying to understand the role of macrophages in wound healing. We know that monocytes circulating in the blood will infiltrate damaged tissue. Whatever organ that's damaged, this happens, including the skin. The monocytes then differentiate into macrophages, and they can perform a number of different functions in the wound. This includes phagocytosis. Macrophages are named for their ability to phagocytose dead cells and debris, so they kind of clean up the wound. They then can also produce a number of different cytokines that kind of help propagate the inflammatory response, but also can produce growth factors important for kind of stimulating the other cells to function to help heal the wound. And also to resolve the inflammatory response. And so this has been a focus for my lab for the last 15 years or so. So over the past 15 years, 10 years maybe, we've done a number of studies looking at the different functions of monocytes and macrophages in wounds. And our lab, in addition to many other labs across the world, have looked at this transition of the monocytes and macrophages from a pro-inflammatory kind of status, where they help propagate the inflammatory response to kill pathogens in the wound, to phagocyte the debris, into another phenotype that's important for healing. So where they produce cytokines and growth factors that stimulates the other cells in the wound to help heal the wound. We did a number of studies in most of our studies are in mice, but we've translated some to human. Found that the impaired healing in the diabetic mice is associated with persistent inflammation that doesn't resolve. We've identified a couple of pathways that have been involved in either propagating the inflammatory response, chronic inflammation, or kind of impaired activity of factors and pathways that help to resolve the inflammatory response. And two that we've studied are the NLRP3 inflammasome, where we see sustained activation of that pathway in diabetic wounds that's associated with impaired healing. And that is linked to impaired activity of a pathway called the PPAR gamma pathway that's involved in transitioning to a kind of a resolving inflammation and healing response. So we've done a variety of different studies using transgenic mice, inhibitors of the pathways, activators of the pathway. You see here, normally you have this ordered transition from a pro-inflammatory to an anti-inflammatory state in the wound when you have an efficient healing in non-diabetic mice. That's at least partly regulated first to pro-inflammatory by the inflammasome and not anti-inflammatory by the PPAR gamma pathway. Whereas in the diabetic mice, you see this prolonged inflammatory response that's associated or in part driven by the inflammasome and the cytokine IL-1 beta, and impaired and lower activity of the PPAR gamma pathway. So this was kind of a traditional approach for studying biology, studying wound healing, looking at specific pathways we think might be involved. But we always had the sense that these studies, analyses were always biased because they came from previous literature that suggested that these pathways might be important, but we never felt like we were getting the full picture of what might be happening in wounds. So I had been very reticent to get into the omics field, big data, for many years, because I just wasn't satisfied that the techniques, the assays available were really going to be able to address the questions that interested me. And this was before the advent of single cell technologies that made it accessible to many laboratories. Very expensive, but when it first came out, really only very specialized labs could participate in this kind of revolution with the advent of technologies that allowed other labs to get involved. That's kind of when we jumped in to use this to try to understand heterogeneity broadly in the wound and interactions between all the different cells in the wound. So these studies first started out by focusing on monocytes and macrophages. We isolate these cells from wounds of mice. We've done some with humans. We do this with traditional cell sorting techniques using these markers. And we look to see how the gene expression patterns of all these cells change over time. So we have in this data set 3 different time points: day 3 after wounding, day 6, and day 10. Here we've got combined look at all the cells generated, and we could see that the cells clustered into 3 early. We're calling these early clusters, primarily pro-inflammatory, dominated by interferon response pathways. Whereas the late macrophages, we call it late because these are dominated by later time points. More healing associated genes are expressed by these cells. And then what we weren't anticipating, but looking back we should have, there's a large cluster of cells that have antigen presenting characteristics. Okay, so typically more associated with the adaptive immune response, if you want to call it that, and not the innate response. But there's a few clues in the literature that these cells could be important in healing. You see down here the transition that's occurring, whereas at day 3, most of the cells are here in these early clusters. This transition is occurring by day 6. And by day 10, the majority of the cells are populating the clusters in what we're calling late. So this kind of gives us our first picture of kind of confirming what some of the data in the literature was telling us, that there was a transition from pro-inflammatory to anti-inflammatory or pro-healing pathways involved. But this gave us a much deeper look in the pathways involved. So collaborating with our data science people, we were able to take advantage of some of the analysis methods that have been published in the literature. One is called RNA velocity. So this kind of takes into account ratios of different states of processing of RNA. It gives you an idea of the trajectory of the cells that are populating these different clusters. And you can see by the arrows kind of a prediction of the transition states that are occurring. We see overall kind of a flow from the early to the late, and some suggestion that there may be some transition between these other populations and the antigen presenting cells. Another technique that's very commonly used with these data sets. It's called pseudotypes. We're trying to order them by kind of the sort of independent of the time they're collected, but the similarity of gene expression pattern between different cells that they use to order the appearance of these different states. And again, showing pathways that could be occurring between early and late. So again, this is not a particularly new idea in the field that transitions occur, but this gives us much greater detail in the transitions that might actually be occurring. We did some experimental approaches to try to validate these ideas by taking monocytes from the bone marrow, administering them directly into the wound, and looking to see if these cells demonstrated evidence of the similar state transitions. And we see a marker we use is LY6C, that they express high levels of LY6C from the bone marrow. They also in blood. Once they're introduced in the wound, they lose this marker. So the black, this is the loss of that marker. And they gain a marker, a maturation marker for macrophages called F480. So you're seeing the state transition that fits with what we see in the wounds. So we think that, we're looking at, the modeling is looking at reality, giving us some results that are real. And LY6C is a marker that's expressed highly in the macrophages in these early clusters, and it's lost later on. Of course, this is distilling all the genes down to a couple of genes, which we kind of need to do to just talk about it, and especially to talk with other wound healing scientists and immunologists. The next thing we wanted to do is try to understand how these different subset of macrophages were regulated. And it's known that transcription factors are very important in the regulation of gene expression in general and in macrophages. And we wanted to be able to use our data to get an unbiased look or less biased look at the transcription factors that might be active in the different clusters of macrophages. So this was again a collaboration with our data science group at UIC, Yang Dice group. They developed this model they call BitFam, which is their acronym for the Bayesian Inference Transcription Factor Activity Model. And so basically what this does is take that single cell RNA-seq data. We also use single cell ATAC data to get ideas of chromatin availability. So, which transcription factors binding sites are available to the transcription factors, which transcription factors are expressed in the data sets. Combine that with some data in the literature that links transcription factors with expressions of certain genes. Use all this to predict the activity of transcription factors that might kind of govern the transcription activity in the different groups. So this says in review there, but we just got notice that this has been published, accepted for publication. So when we look at the different subsets of macrophages, so we have three different subsets of the early macrophages represented here, one of the late and one of the antigen presenting cells. And we see that there are different transcription factors predicted to be active in the different groups. Some of them make a lot of sense. So for example, in the late, MAFB has been very well studied in the differentiation of macrophages. So monocytes to macrophages makes sense that it's active later. STAT2 is a very well studied in inflammatory responses, interferon responses. So it makes sense that that's active early. And then there's a variety of other transcription factors that, again, studying the literature makes sense, and others that were kind of new or maybe a little more controversial, including this one NR4A1 that's been described in the literature as both pro-inflammatory or anti-inflammatory, depending on the context, right? And so those kind of things are always very interesting to me to see, okay, in our system, how is it acting? So this, to me, was a good example of being able to use these kind of data sets to generate information that was much less biased than what we've done before in terms of picking one and seeing what kind of role it plays in our process. We've got a larger picture view here. So next, they did some network analysis of these different differentially inferred activity of the transcription factors. See how these group together. We see some here that were mostly in the late macrophages, more here that were in the early. And again, some made sense, some are kind of surprising. So we follow those up with some validation experiments. A small community here included NF-kappa B, which has many functions, but pro-inflammatory is kind of one of its main, and it was linked with NR4A1. So this gave us some clue that maybe NR4A1 in the wounds was playing a pro-inflammatory role. And so that's kind of what we followed up for validation in this analysis. So, I thought I put in new data, but it's not there. It's funny how the different versions of this anyway, we can talk through it. So here's some of the new data that we generated validating the NR4A1. We see that the protein, all that data that all the analysis before was based on RNA data, message RNA, and chromatin availability. Here we're looking at protein by flow cytometry. We see this protein is expressed more highly at early time point than late. Again, consistent with an idea in a pro-inflammatory state. We see a modest but significant decrease in expression of pro-inflammatory markers in the NR4A1 knockout mice, macrophages from those knockout mice. We've done some other studies with bone marrow transfer between the NR4A1 knockout mice and a competent NR4A1 competent mice wild type mouse. And we see that when you knock out that gene in bone marrow cells, you see a reduced initial inflammatory response. So we got nice validation that the model predicted this factor might be involved in pro-inflammatory, which was processes, which was not 100% gimme from just those data, and the experiments validated that it seems to be playing this important role in the pro-inflammatory response. We got some pushback from the reviewers because of this fairly modest reduction in pro-inflammatory cytokine levels, expression mRNA levels, but to me it kind of made sense. I mean, I'm not expecting that we're losing expression of all inflammatory cytokines by getting rid of one transcription factor. We think a lot of redundancy, a lot of things that are playing a role to get some significance at all was very exciting for us. We're continuing on with these validation experiments. There's unlimited validation experiments that we can do, which take a lot of time and a lot of money, based on all the great information that we have from the prior analysis. So the next step was to compare the macrophages between the normally healing wounds and the poorly healing wounds. So on top is the macrophages from non-diabetic mice, bottom is the macrophages from the diabetic mice. And we see again here transition from these early macrophages to late macrophages in the wild types. And again, the APC related cluster out there on the right. And you see at each time point in the diabetic mice, the populations look very different. You don't see, they start out different, and you don't see the transition. They look more similar between time points. So this is kind of consistent with this idea that inflammation is dysregulated in diabetic wounds, and you don't see these state transitions that we normally see in a normally healing wound. The other interesting thing was there seemed to be reduced numbers of these cells with the APC phenotype, which was interesting as well. So we kind of are following up. There's lots of different things to follow up here with all the different subsets, but one that has really not been described in wound healing are these cells that have the APC phenotype. This is the cluster here, and if you look at the population, non-diabetic is the majority, whereas diabetic is much fewer. We see that these cells do express a number of markers typically associated with antigen-presenting cells, dendritic cells, if you know any immunology, dendritic cells or macrophages that have antigen-presenting capabilities, and they express lower levels of more macrophage-related markers. So we're pretty confident they're some kind of dendritic cell or macrophage with the dendritic cell capabilities, maybe. We, again, validated this at the protein level with flow cytometry, and we're seeing this is kind of the gating strategy we used, focusing on the markers that we identified in our single cell data set. And again, we see accumulation of these cells. Well, it's very challenging to actually find the right markers for flow cytometry protein levels that fit our single cell RNA data, but this was our best guess right now. And again, we see accumulation of these cells in the non-diabetic mice, and they are reduced in the diabetic mice, which is consistent with our RNA-seq data. And looking at different expression levels at the protein levels, different markers, typically highly expressed other markers associated with these types of cells and express lower levels of macrophage-specific genes. One thing that was very interesting is there's a marker called CD301 that was very different between non-diabetic and diabetic at each time point. This is something that we're going to be following up to understand the function of that protein and whether it's playing a role in the chronic inflammation and the impaired healing in the diabetic wounds. So to understand kind of where these cells are coming from, there's an idea that if they're true dendritic cells, then they come from pre-dendritic cell precursors. And if there's a potential for monocytes to differentiate down this pathway to become more antigen-presenting cells. A fairly common mouse that's used to study kind of monocytes in mice is the CCR2 knockout mice. So in CCR2 knockout mice, monocytes accumulate in the bone marrow and they're deficient in the blood. So they're produced in the bone marrow, but they don't get out of the bone marrow into the blood. And so you have a deep deficiency in the blood. And if you have deficiency in the blood, you'd expect to be fewer of monocyte-derived cells in the wound. So monocytes will infiltrate the wound and differentiate into macrophages, or we think into dendritic-like cells. And in these mice that are deficient in blood monocytes, we see significant decrease in these cells with ABC characteristics. We use various markers. CD209 is one that's expressed very highly by these cells. Again, see a deficiency in those cells. And so we think that at least the majority of the cells in that ABC cluster are monocyte derived. We did another type of experiment to validate this, where we did bone marrow adoptive transfer. So sorted monocytes from bone marrow, introduced them into wounds of either non-diabetic or diabetic mice, and looked to see how they differentiate in the wound. We see that, again, evidence that they differentiate into the ABC phenotype cells in non-diabetic wounds, but in diabetic, there's a kind of impaired differentiation, reduced differentiation. So we think that the wound, the environment of the wound is contributing to this impaired differentiation, because they differentiate into this phenotype in non-diabetic mice, but not in diabetic mice. Whether this is where, it has an impact on healing and inflammation in general, those are experiments we're still doing. And I think that's kind of, this is just a slide to show that it's even more complex than all of that. That even within this IPC cluster, there's different subclusters that seem to express different levels of some of the markers. Particularly one that expresses high levels of CD24A, and one that expresses very high levels of higher levels of CD64. So different markers used to identify macrophages and dendritic cells. When you use flow to identify these different populations, again, there's one subpopulation where we see that trend again for reduced numbers in diabetic wounds, but then others that show increase in the diabetic wounds. So we're trying to tease all this apart to try to understand the heterogeneity within the heterogeneity, which is very challenging now. Experiments that, again, my postdoc and my research assistant professor are, I'm going to skip this one. I took it out. So, to conclude this section of the talk, the macrophages in diabetic wounds seem to show kind of impaired state transitions, this kind of persistence of inflammatory phenotype and impaired accumulation of these ABC-like phenotype cells, that those latter cells are derived from monocytes, and monocytes differentiation to dendritic cells, the typo there, is impaired in the diabetic wound environment. And we're using different strategies to target these cells now, try to deplete them in the wounds to test their function, to see what influence they have on inflammation and healing. And those data are, it's kind of a cyclic process. Target, see if what happened, you think the cells that you're targeting actually get depleted, and then see what that impact that has on inflammation and healing. And too many surprises, not ready to talk about them yet. Okay, so we're going to move on to communication between the macrophages, which has taken up most of the talk so far, and other wound cells. So we have another whole set of data that is looking at all the dermal cells in the wound. And this includes the macrophages here. We see the total population of macrophages. Our ABC cluster is out here. We have lots of neutrophils, bunch of fibroblasts, endothelial cells, smooth muscle cells, and NK and T cells. So, our immune cells are all here, and then other cells in the wound here. And it's known there's a lot of communication taking place between the different cell types, but exactly how this is all working together and is coordinated is very poorly studied. Usually you pick one cell type, another cell type, and see if you can find some evidence of communication, but trying to do this all together has really not been possible until the assays have become available, and then the informatics approaches have become available to integrate all of these things. So the first thing we notice is that if we look at populations of macrophages between non-diabetic and diabetic mice over different time points, that these are different. You pick any cell subset, you can see these differences. The neutrophils are very evident. They cluster together, but they've occupied very different parts of the cluster, and this is sustained throughout time. You can look at the fibroblasts as well, very different phenotypes that are expressed. And so we have different people we're collaborating with that are, one guy is very interested in fibroblasts, so he's really looking deeply into that. Another person with the endothelial cells, and so on and so forth. The nice thing that, again, you always want to try to make a check with reality that even at this levels, we're seeing things that are consistent with literature. So we see reduced numbers of endothelial cells in diabetic wounds. There's impaired perfusion of wounds, and this is very well known in diabetic wounds. We see, in general, reduced numbers of fibroblasts with a different phenotype, so on and so forth. So we think we've got a nice data set to work with. Again, different experts in different fields have said, "Oh, you got a lot of fibroblasts, and this is great." It's good data, and so it makes us feel good that we did it the right way. So there's a package called CellChat that was developed by investigators at UC Irvine that kind of predicts cell-to-cell communication that we're interested in using levels of expressions of known ligands and receptors on different cells. As well, they try to incorporate in their model, receptors that have more than one subunit, as well as modifiers. And so we run this package. It comes up with lots of different analyses that can be visualized in different ways, and we're still kind of working through this. The nice thing is that we see early on, for example, we see high expression or predicted activity of inflammatory pathways that you'd expect. So those involved with CXCL pathway, the MIF or CCL. So these are all highly predicted to be active early in wound healing, which makes sense. And we see more predicted interactions in the diabetic mice than in the non-diabetic mice. And so again, this is consistent with what has been reported in the literature. And so we're pretty confident that this is sort of making sense. There's a lot of new things in here. Some of this SPP1 is called osteopontin. It's been studied in wound healing to some extent, but not in diabetic wound healing. It's thought to be involved in scar formation during normally healing wounds, but it seems to be upregulated in diabetic wounds. Also has a pro-inflammatory role. So we're going to be looking at its potential role in kind of propagating inflammation in diabetic wounds. Here's some examples of other ways to visualize the data. So you have all the different clusters here, non-diabetic mice, diabetic mice. Cluster 0 are the macrophages. Cluster 3 here are the neutrophils. CCL signaling is a chemokine, very important for attracting inflammatory cells into the wound. And we see the major kind of players in producing or expressing CCL are neutrophils and macrophages, which makes total sense. And it's interesting that this package predicts that there's more interactions in the wounds of diabetic mice, that more kind of fibroblast populations are interacting in this pathway. So there's a lot to follow up with that, because there's a couple of these cytokines that are pretty well studied in wound healing, but this is suggesting that a lot of different family members of that pathway are involved. And I'm not even sure how to study this experimentally. VEGF signaling is another one that's very well studied in wound healing. It's known to be impaired in wounds of diabetic mice, and we see the strength of the interaction is predicted by the thickness of these lines. We see the line much thinner, so predicted to be much less strong in wounds of diabetic mice kind of during this day 6 period, where you see a lot of angiogenesis. This pathway is involved in angiogenesis, new vessel formation in wounds. It's very strong in non-diabetic mice, impaired in the diabetic mice. And it's known that angiogenesis, new vessel formation in diabetic wounds, is impaired. So these make some sense to us, these predictions, and some of them we're going to try to follow up on, because it gives you predictions about different family members that might be involved in the actual signaling pathway. This is kind of another view of this one specific pathway called osteoponin. When we look early, we see that the thickness of the lines much bigger in the diabetic wounds compared to non-diabetic, suggesting this pathway is more active, especially involving neutrophils and macrophages, signaling to producing osteoponin, or at least expressing it, and potentially signaling to receptors on lots of fibroblasts and endothelial cell populations. When you look at it in this way, this is outgoing interaction strings, so cells that are producing the ligand. This is the incoming interaction string, cells producing or expressing the receptor. And this is all differential between diabetic and non-diabetic. You see SPP1 is kind of one of the outliers that's very differentially upregulated in diabetic wounds, particularly interacting macrophages with other cells. So this, and then on the right here, you see SPP1, that's osteoponin, that's the ligand, and potential receptors that are expressed on the fibroblasts and other cells that it may interact with, and showing the differential interaction between, in the red, non-diabetic, and in the blue, the diabetic. So this is a pathway that we're going to follow up with kind of experimental validation. It has not been studied in diabetic wounds, and could be kind of one of the drivers of the inflammatory process in the diabetic wounds. All right, so to tie up that part, we're looking at the communication between our favorite cells, the monocytes and macrophages, with other wound cells. Lots of different pathways they think that are predicted to occur. A lot of them have been kind of reported before in the literature, some of them have not, and again, validation experiments going ongoing. So it gives us a rich source of targets to follow up, more than I'll be able to follow up in a lifetime. So the last thing I want to talk about is what kind of brought our interaction, Christina and I interaction together, looking at mechanical stimulation and wound healing. My training from way back is engineering. I've always been interested in how mechanical signals influence cells. And so I had drifted away from that. This project kind of helped me bring me back. I was collaborating with a group at Stony Brook University in New York who were looking at the impact of vibrations on bone. And they were using these very low intensity vibrations to try to ameliorate bone loss, osteopenia, or kind of disuse atrophy. And they were getting some really nice results from that. So we decided they sent us some equipment. Let's see what happens with wound healing. We had some students. They needed a project. So let's have them start this and see. I didn't have a lot of hope for the project at first, but undergrad students need something to do. It's something they could do, get some experience with research. And the first set of experiments seemed like it improved the healing. Didn't really believe this could be possible, that it was happening to the degree it was. So I had another group of students work on it. Same results. So we started to believe and had a postdoc work on it. Same results. So that kind of launched this investigation into what can happen. Now, there's a lot of skepticism about kind of how this could be having an impact on healing. Most people think this is kind of magic. We have this machine here that, I don't know if this was popular in Italy, but in the US, back in the 1960s, early 70s, this vibrating belt that you go, was it?
To lose weight.
To lose weight, yes, exactly. And you didn't have to do anything. You just sit there and lose weight. And so, the science community is very skeptical, to put it mildly. But there were some studies in the literature showing that these vibrations could improve perfusion in the skin. So you start the vibration, different parts of the body, you can measure increased perfusion, blood flow through that region. And it's been reported many times. Because perfusion is so important for wound healing, we thought, well, that kind of makes sense that this could be occurring. So some data. So these ideas go back to Nikola Tesla, the American inventor, born, I don't remember where he was born originally. But he published a couple of kind of anecdotes about mechanical vibrations and their influence. And apparently he was using this to treat his friends. Like this one, affect the condition of the stomach, influence on the liver, affect the glands in the limbs, kidneys, and bladder. He cured a number of people, including his friend Mark Twain. I don't think there was any data associated with this, but lots of stories. So we include little quotes like this when we try to get funding. So our first studies were looking at healing of wounds, and so you have the controls and the vibrated mice, and we see that the wounds close faster in the mice that get vibrations. In these studies, all we're doing is taking a mouse, putting it in an empty cage that's sitting on a platform that vibrates. And the vibration, we're finding 30, 45 hertz is a good frequency. the amplitude is only a fraction of a millimeter, maybe 0.1 millimeter, and the G-force is less than 1 G. So it's not unlike what happened when your cell phone vibrates. Very similar to that. And we see when we measure histologically, we have faster closure. We have more tissue underneath that also forms.
And sorry, and
Sure.
the wound is on the back.
Yes. It's on the back of the mouse, it's right on the back, right? And so they're walking around. This is why I was originally skeptical. It's like they're walking around, mice are doing whatever they want to do. They're awake. So, yes. The other thing that we see is happening, this is a staining for endothelial cells for new blood vessel formation. We see more vessels being formed, endothelial cell probably proliferation, M2 formation occurring in the granulation tissue under the wound. This is associated with increased levels of various growth factors. VEGF is one I talked about before, higher with vibration. IGF-1 important for proliferation of various cells in the wound. Not all cytokines were increased. IL-10 was not, IL-6 was not, so on and so forth. We've done a number of different studies in this, showing one. Here we looked at different vibration protocols. Different combinations of accelerations and frequency. And interestingly, here we found that of all the combinations we tried, and you could try infinite numbers, there was one combination that improved healing, the others did not. And this was the one we called LL, so it was our lower amplitude and lower frequency, was improved healing or closure of the wound. The high intensity vibration protocols actually delayed closure. It makes some sense that they're at a point of high intensity vibrations, you're going to impair processes. People that work with vibrating machinery can have a lot of problems. People with jackhammers, they have to get impaired circulation of the fingers. It's called white finger syndrome or something like that. So there seems to be kind of an optimum protocol that can be used to improve healing. We've done a number of studies that I'm not going to talk about that kind of target specific pathways that we thought might be involved in healing. But again, this vibration can probably affect lots of different cells and lots of different pathways. So we went back to single cell RNA-seq to try to get this broader view. Again, we can identify all the various cell types in the wound, at least in the dermis of the wound or the granulation tissue, fibroblasts, endothelial cells, macrophages, T cells, neutrophils, and some minor cell populations. And then we can see kind of how the vibration affects these cell populations. We don't see a lot of effect early on, so basically after one vibration protocol, but we start to see bigger changes when we get more bouts of vibration later in the healing process, and that kind of makes sense. Again, these data we're still kind of moving through. We're looking at differential cell communication again in these pathways in terms of vibrated and not compared to not. You see that early on there isn't a big difference between in the total kind of communication that's predicted to occur. But later on, we're seeing some evidence that the total communication might be increased by the vibration stimulus. Again, we're looking at more specifics here of the different cell types that might be involved early on and late. And we can see mostly later on that macrophages, neutrophils, primarily the red means increased interaction with vibration condition, and these are kind of the groups that look the hottest to us in terms of changes with vibration. And I will present one story here that we found interesting. This is a pathway that we think could be playing a role resolving inflammation in the wound. Vibration is kind of increasing. So here we see this annexin A1 pathway. Looks like it's increased. The thickness of the lines increases with vibration on day 6. Neutrophils are the big target for this pathway. Lots of other cells produce annexin A1. FPR is the receptor. And so this is potentially going to have different functions, but anti-inflammatory role is one of the functions. And we see that vibration increases expression of annexin A1 in macrophages and fibroblasts, and it increases expression of the receptor FPR2, especially day 6. So we have increased expression of both the ligand and the receptor of this pathway induced by vibration. And well, again, it's possible it could be playing an anti-inflammatory role. Now, experimentally, we need to try to validate that. Again, one of many stories that we can tell with this data set. There's a beauty and the bane, I guess, of all these data. So to summarize that part, low intensity vibration improves angiogenesis, formation of granulation tissue, and closure of the wounds, reepithelialization in diabetic mice. It looks like we know that it reduces neutrophil accumulation in the wounds as well. Didn't show that data. And one of the ways that it might do that is by increasing communication between other cells and neutrophils involving an annexin A1 pathway. And this is thought to both induce apoptosis of neutrophils and kind of prevent migration. So it could have an anti-inflammatory role in that way. And validation experience ongoing. I think we can end there. And thank you for your attention. And I'm happy to ask any questions.
Let me first check if we have questions here. Okay. Are there questions from the audience?
Sure.
A question on the methodological question on the single cell data set. So you have a time series, and each experiment has a population of single cells. And at the end, when you show the pictures, you have this subpopulation which is invariant, but it should be the subpopulation of the overall experiment, day 1, day 6, 3, and day 6 cell. You have just one big population of cells, and I imagine.
Yeah, so we do.
Because you have a single shape, and
Yes.
you put together
Yes.
day 1, day 3.
Yes.
day
So
6.
we analyze, we've done it a variety of different ways, but we do combine all the data into one data set first, and then try to see if there is kind of a sample to sample bias within the data sets. But we see overlap of lots of the cell subsets, so we think that it's comparable for the most part. But yes, so then the advantage of doing this by combining them and then kind of clustering and identifying cells is that you can then identify for each cluster, which samples are contributing to each of the clusters. So that's the way we've done it. I don't know if that answers your question.
Yeah, see, in fact, there is another part of the question is then when you build up with this huge population, then you have to perform the pseudo time or the
Yeah.
low
Yeah.
dimension, lower dimension
Yeah.
algorithm. I don't know if you use maybe TS.
Yes, yeah. So
TS.
PCNA, and then we do the I don't remember. We do Disney, yes, but then there's the other one. Yeah, just usually we try to look to see which kind of components, the principal components, are kind of contributing to the information and cut off at a certain point. And then we plot various ways to try to see which gives us the best representation, but it's hard to, I don't think there's any one way to decide which is the best way, right? To visualize this n dimensions into two. So I'm, again, not sure if I'm answering your question or.
Yes, okay. I understand that you reduce on the
Yes.
on the overall population, and then when you do pseudo time, again is a lot to reduce dimension of data and to find some part of. There is a correlation with the real time that you have.
Yes.
So you recognize that the cell express the expression of cell at day one corresponds to the beginning of the path individuated.
Yes, yeah. So we tried to compare with reality as much as we could, and it makes sense that the cells that we would identify as kind of root cells, or whether it was RNA velocity or pseudotime, were kind of the early stage, what we call the early stage cells that were mostly populated by the early time points. But there's always a distribution at each time point that even within the early stage day 3 wounds, there were some that were kind of already in what we were calling the later stage wounds, and the path was kind of following this. It's known that there's this kind of distribution of differentiation within wounds, within tissues in general, in a healing process. And the idea is, pseudotime is trying to capture that and account for that.
There is a coherence.
Yes, yeah. For the most part, yes. And then it works very nicely in the normal healing wound, but diabetic wounds things are happening very differently. How best to describe this, I think, is still kind of a challenge. Like, is it just, most of the biologists are describing this just as kind of a stopping point in the transition. But it looks to us like the starting point is different too. And they don't move very much from that point. So, yeah, so they're again, as much as my understanding allows me and our techniques are available, we try to validate and make sure that it fits with what we know in terms of time, in terms of what's in the literature.
I have a very basic question. When you perform a single cell experiment, you 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 look just at one kind of cell like monocyte, or you have an ensemble of different type of cells?
So we've done both ways. So we've the first data sets that I talked about, we sorted out cells that we thought were monocyte macrophage type cells by using specific markers that are pretty well accepted. So we sorted and then captured single cells and
Analyzed
right.
precisely.
Yeah. The others, the later data sets I showed was taking the entire wound, scraping off the top, which there's a lot of junk, and we get rid of, what some people are very interested in, the epithelial cells that help for the wound, but we're getting all the different cell populations then. So we, the technically challenging part of this is going from a wound or any tissue and separating the cells, right? And the impact that could have on the data. But yes, so that's kind of that's where we want to move our work and our thinking, because the interaction between those cells is what is healing the wound.
More questions? I have one about time. Can I ask? And then so I think that something that was very interesting was that this early time point was somewhat shifted to day 3. Normally, the very early stage of wound healing are considered between 20 and 48 hours.
Yes.
So that's very, very interesting that when you go into the details, in fact, it lasts a little bit longer.
Yes. These experiments cost a lot of money, and so you have to make choices about which time point you want to use. We did use the day 1 for the vibration because I wanted to get the capture right after the first one. But it would be interesting to go even earlier and see, especially with difference between non-diabetic and diabetic. Are we seeing differences right away?
But it's still very clear what
Yes.
I mean, between day 3 and day 6, it was
yes.
obviously a different stage.
It seems like, again, biology doesn't always happen super cleanly, and we've done other experiments where the day 6 wound looked a little bit more like the day 3 wound. But we think that big transition occurs somewhere in that time frame. Yeah.
I have more, but your turn.
Did you perform a gene regulatory network inference from this data?
So
No, I don't think so. Maybe, I mean, the network I saw, this was co-expression network. And this is the only network approach.
So I'm trying to think if we've done. I don't think we've done this yet. There's a lot of things that my collaborators, students doing, and I don't think they've done GRN.
Yeah.
Okay.
So the last question, data are available for others? Or
What's that?
Your data set are
He wants
available?
your
Yes, so we have published the non-diabetic mice data is publicly available. It's already shared. The ATEC-seq data, so the chromatin availability is, we just had that published. The diabetic mice is not public available, but I'm happy to share if you are interested.
Okay, thank you.
He wants to do the inference.
Okay. Yeah, we're happy. I mean, the beautiful thing about these data is we're pretty confident that we have a high quality data, and I won't be able to do all the things that could be done ever. So other people that we're happy to work with. Sure.
I have a couple of questions. For this NR4A1,
Yes.
has this been tested as a drug target, or
I
do
think
you
it-
know?
Okay, thank you. So it's also called NER77. There was a few studies done with atherosclerosis. and people thought that it was, there was, studies on both sides of
Yes.
the coin. I don't know if if it's targeted, then it's most likely cancer, or cardiovascular disease.
Okay, but not
I don't
as
know if it's
as anti-inflammatory.
yeah,
probably
I
not.
don't see the problem is of developing the target or drug is that most people think we're falling on the side that it's anti-inflammatory, so you wouldn't target try to inhibit
Yeah.
it, right, in that case. But who knows whether this is really context specific or what. So yeah.
I have a couple more questions.
Okay, sure.
One is about a technical question for the leaf, the vibration.
Yes.
Did you do it just once and then you sampled, or
sorry.
you repeated it?
It was repeated every day.
Okay.
Yeah, so it's 30 minutes a day. For these studies, it was all whole body.
Okay.
We've done some studies where we have to anesthetize a mouse and we just shake near the wound.
Okay.
Yeah.
And it works better?
It works. I don't think we can say it works better. The nice thing is that
It works.
it works.
Yes.
And some of the kind of phenomena are kind of consistent of local versus whole body. The pathways involved, we haven't looked at all of them, but some of them I think are activated by local and others are not. So there's some differences, but yeah, it's hard to say.
Yeah, because on a translational perspective, of course, it would be better to have local vibration.
Yes,
than...
yes, and that's the whole purpose of that. We're trying to develop a kind of a buzzer, similar to what you have in your phone, that you can apply kind of in a bandage and control by a phone, basically. It could be very simple, cheap way to address this. So, yeah.
Last question. You mentioned this pathway of annexin A1.
Yeah.
So if, I told you we have reconstructed this mechanotransduction pathway, but we don't have a stimulus, the low vibration. We have different type of mechanical stimuli. If this pathway is, it's already a good starting point to attach this to the map and see what happens. So this could be very interesting to.
Good. Yeah. And again, if you always time and resources, but we have the data set. So if you were looking at exploring more globally, we'd be happy to share that.
Any more questions? Okay. I think here also from the audience, we're okay. So thank you again.
Thank you. All right. Thank you.